I'm Takashi Yamashiro from Osaka University. I would like to say thank to the organizers for giving me a chance to talk at this AAO meeting. Unfortunately, I still have some difficulty to travel abroad from Japan, and it is a pity that I cannot show my presentation in person. Today I'm talking about our 3D cephalometries and how we develop and applied it in our clinics and research. And this is my COI, and I have the following financial relationship to disclose about my research funding and patent applications. And this is the topics. And today I'm talking about the limitations of the 2D cephalometries and how we developed our 3D cephalometries. And I'm showing how we evaluate the 3D image of the human and the experimental animals. And I'm also showing how AI and machine learning are applied in 3D cephalometries. And in traditional 3D cephalometry, the skeletal deformity was assessed by linear and angular measurement. And so the problem is that the 2D cephalogram is based on the too much simplification of the anatomical structures. As you can see in the pictures, the 2D pictures does not illustrate the complex feature of the craniofacial structures. On the other hand, now the CT or the ConBeam CT is available. And the CT provides a three-dimensional image of the craniofacial structures with high resolution, which enable the details and the direct observation of the craniofacial structures. And however, the many previous studies using the 3D CT performed the 2D observation on a reconstructed slice, or the measurement among the reference points and placed on reconstructed models. But this limitation was improved by the application of the semi-transparent overlays approaches. And this figures is the previous studies of the Hino et al. And they evaluate the effect of the bone-anchored maxillary protrusions, bump, and three-dimensional displacement between the pre and the post-treatment was clearly visualized as the gradient of the color code on the 3D images. But however, the problem of this approach is that the data is too big. And so the treatment effect was not averaged. The average cannot be calculated on these studies. So our challenge was to optimize the data size, which enabled to calculate the averaged values and establish the standard three-dimensional models. And so we described how we developed the 3D cephalogram. The idea is simple, and we are using wire meshing fitting to capture the 3D shape. In these pictures, wire mesh was fitted on the bottle surface, and the right panel of the bottle is represented by a 3D wireframe consisting of the triangulated mesh. And this wire mesh provides the 3D coordinate values on each point, and which reduces the size of the morphological data without compromising the quality of the 3D imaging. And I'm showing how we can manipulate the convexity data. And this is the original and raw CT image in DICOM format. And from this data, only the surface data was extracted as STL data using this software. And at this moment, the patient sample data have the 1 million points. But it is too huge to be manipulated, so we have to downsize the data size. And we are also showing the image of the bottles here. And in order to reduce the size, we prepare a template mesh with 10,000 points. And using this reference point, this template is fitted to the patient models. So in this process, we used 23 and 29 landmarks in the maxilla and the mandibles respectively. And accurate landmark identification is critical for the reproducibility of the surface-based models. So after the template mesh was fitted on the patient models, the homologous patient model is generated. So that this 3D standardized wire mesh image has just 10,000 points. And now we can generate the averaged image. And using these approaches, and we generate average the three-dimensional model of the 10 years growing human samples with skeletal one relationship. And the sample feature is shown in this table. And this picture shows how the average model is generated from the sample. We prepared 28 sample data. And each sample have the 10,000 points. And at each wire mesh points, and the standard deviation were calculated on each X, Y, Z axis. So the average model have the 3D coordinate values and with SDs. So the color code of the value can be mapped. And so this color code means the 1 SD values. And this 1 SD value was mapped on these 3D images in X-axis, Y-axis, Z-axis. So this figure is a little bit complicated, but the value of the 1 SD was mapped on the 3D image as a color code. And as you know, the 1 SD is the normative ranges. And so as you see the color code, the blue or the gray has the small values, and the pink or the red is the high values. And high value means the so much variations. And these range were mapped in each X axis, Y axis, Z axis. X axis means the transverse, Y means the vertical, and Z means the anterior posterior directions. As you can see, these pictures, and in contrast to the maxilla, the color of the mandible is more the pink yellow, especially in Y-axis. So this means the normative ranges in the Y-axis in the mandible is so much. And in general, the variation is much more in mandible than the maxillaries. And of course, we calculate the reproducibility of the wire mesh fitting, and reproducibility was confirmed by the intra and inter-observation reliability, and we calculated the ICCS, the intra-class correlation coefficient, and MDC 95, and 95. This means 95% confidence intervals, and both values shows this analysis has a good reproducibilities. Then I'm showing how to evaluate the 3D craniofacial image of the human and animals. And this is the 3D image of the Loeys-Dietz syndrome patient. And I'm showing how we can detect the skeletal phenotype of this syndrome. And as you can see in these pictures, this patient shows, for example, the midfacial deficiency, and notch is evident here, and this patient shows a high mandibular plane angles. And we can detect these phenotypes on the 3D images. But anyway, this picture shows the superimposition of the case and contour, and the case is shown yellow and the contour is shown blue, and the skeletal displacement is evident. And as I explained, these models, each model are wire mesh with the 3D coordinate values at each point, so that we can calculate the amount of the actual displacement, and we can also calculate the significant difference values at each point in each X, Y, Z axis. And so this figure shows the significant difference map. And the significant difference map visualize the skeletal displacement as a color code. And this color code indicates the SD values, Z-scores. So this SD values, Z-score was mapped on the 3D images. So this blue colors, dark blue colors, and this dark blue indicates the low SD values. So this means the backward displacement of the anterior portion of the mandibles. And this yellow color in the condyle on the conoid process indicates a major displacement of the condyle of the conoid process. And in these directions, AP directions, anterior posterior directions. So this figure is also the significant difference map of the maxilla in the AP anterior posterior directions. And this dark blue indicates backward displacement of the anterior portion of the maxilla and the craniofacial bones. And this pink indicates a major displacement of the temporal bones. And this figure is also significant difference map, but in y-axis. Y-axis means vertical directions. And the pink color here indicates the marked downward displacement of the infraorbital region of the maxillaries. And the red colors of these regions indicate also the marked downward displacement of the mastoid process. And this yellow means the downward displacement of the upper teeth and the surrounding alveolar bones. And so this is the summary of this patient in each X, Y, Z axis. And anyway, the significant difference map clearly illustrates the area of the displacement of the patient. And the 3D cephalometry provides more detailed finding of the skeletal displacement. Anyway, I'm also showing the other examples in animal studies. And it is established that the soft food diet affects the craniofacial development. But such a previous study was based on the 2D radiograph, and the findings are based on the linear or the angular measurement of the reference points. So we reanalyze the skeletal phenotypes of this in this experiment. And the data processing is similar to the human studies. And original CT data are obtained by the micro CTs for the animals. And its resolution is also similar to the Conbeam CT, and template mesh was mapped on the samples. And the template mesh was, in this case, is about, this is also similar as a 10,000 point. And so the sample models with the about 10,000 mesh point was generated. So now we can generate the average model of the control and the experimental animals. And this, in this case, we had the 5 soft diets and the 5 control animals, and we can have the averaged soft diet experimental models and averaged control models. And so that we can compare these 3-dimensional models. And the process in this studies, the skeletal displacement was quantified by the actual displacement and the significant difference at each point. And those values were mapped on the 3D image as color codes. And this left panel is the actual, this color code of the left panel indicates the actual displacement. And this right panel indicates, this color indicates the significant differences in each point. And this figure shows the significant difference map in each X, Y, X, and Z axis. And all the significant displacement in each direction was mapped on the sample 3D image of the mandible and the maxillaries. And unfortunately, today we do not have enough time to interpret all the findings in detail. So, and we are picking up the finding, and this is the short summaries. And anyway, this picture illustrates the significant difference in vertical directions. And this violet color indicates the significant upward displacement of the lower, the posterior lower border of the mandibles. And in the zygomatic arch regions, the downward movement was also evident, as well as the temporal bone regions. And this is the summary of the significant displacement in the X transverse displacement. And it's very interesting. This could be hardly detected, but in coronoid process, and the significant inward displacement was evident here, and lower molar region also showed significant inward displacement. And this is maxilla, and the diagrammatic arch also showed the inner displacement in the soft food diet mice. And the temporal bone regions also displaced inward. And this is the displacement in the anteroposterior displacement. And in mandibles, these notch regions moved in these major directions. And in maxilla, the anterior portion of the diagrammatic arch, basal portion of the diagrammatic arch moved backward. And in contrast, the temporal bone regions moves anterior directions significantly. And it is very interesting. This color distribution indicates that the significant skeletal displacement occurs at the muscle attachment region of the diagrammatic arch and temporal bone and the mandibles, suggesting that the musculatory dysfunction affects the skeletal development in the growing animals. And since the skeletal phenotypes in those areas are hardly illustrated on the 2D X-ray image, and so we may have to overlook the essential finding and the phenotype in the evaluation of the soft food diet mouse. And this is our summaries. And we have shown the great potential of the 3D cephalometry in both human and experimental animals. And however, 3D cephalometry is very complicated in interpretation of the finding and also in data processing and landmark and identification. So we are afraid that the audience hesitate to do this. So we apply the machine learning and AI in data evaluation and landmark identification to facilitate our process and data analysis. And this is the final topic, and I'm talking about the application of the AI and machine learning on the 3D cephalometry. And anyway, the craniofacial morphology have the variations, and this variation is influenced by the genetic and environmental factors. But it is not easy to interpret which factors significantly influence the craniofacial characteristics. So in order to extract the explanatory factors in adult craniofacial bones, we evaluate the craniofacial characteristic using the principal component analysis and the PC clustering. This is one of the methods of the machine learning. And this is a material method, and I used the 60 Japanese adult patient with skeletal malocclusions, and the 3D surface models are constructed from the Conbeam CT using a wire mesh fitting method, and superimposed by the Proteus registrations. And the explanatory factors of the morphological variations were extracted and analyzed using the PC analysis and principal component analysis and PC clustering. And we also evaluate the sexual dimorphism. And in these studies, ESCAL approval for these studies was obtained from our university board. And these are the results, and we found 9 principal components explained 62% of the morphological variation. And the right figure shows the principal component, and yellow is the over-exaggerated images, and blue is the under-exaggerated images, and both images are overlapped. And today we do not have the time, so I'm explaining the principal component 1 and 2. And as I said, the yellow is the over-exaggerated image, and the blue is the under-exaggerated image. And the deviation is evident here. So and from these features, we can say that the PC1 is positively associated with the anterior divergence of the mandibular bodies. And PC2 is the difference is evident here. So the PC2 is negatively associated with the anterior facial height and accompanying the shape of the gonial angle and the protrusion of the upper incisors. And from this principal component, we found the 4 clusters were found to be optimal to represent the sample variation. In other words, we can say that in Japanese adult, and we have the 4 types, and type 1 have the high mandibular plane angles, and type 2 have the skeletal 2 tendency, and type 3 have the brachyfacial and with the short facial height, and the type 4 have the bimax protrusions. And this is the result on the sexual dimorphism. And we also found 3 principal component account for the sexual dimorphism. And these principal component associated with these skeletal features. And the combination of the principal component 7 and 3 successfully separated the male and females. And also the combination of the PC7 and 5 also separates the male and females. And this is conclusions, and 9 explanatory features were extracted, which explain the 60% of the craniofacial morphological variations in human adults. And we found 4 types in Japanese adult. And we also found the 3 principal components account for the sexual dimorphism. And we can also say that machine learning supports interpretation of the 3-dimensional big data analysis. And this is another approach. And we also developed AI supporting automatic landmark identification. And we have already evaluated the reproducibility of this automatic identification, and I hope we can show it at the next meeting. So this is the conclusions, and we developed the surface-based analysis of the 3D image to quantify and visualize the craniofacial bones morphological features. And 3D analysis provides the more detailed feature of the craniofacial morphology than traditional 2D analysis. And machine learning supports the interpretation of the data analysis of the 3D digital big data. And AI proposed the automatic landmark and the identification in 3D craniofacial images. And I would like to say to our colleagues, and Chihiro Tanigawa she has done the 3D analysis, and she is also the specialist of the AI and machine learning. And Yuka Murata and Kono of the Okayama University has done the animal experiment on the soft food dieting. And the Hiroshi Kamioka and me also organized the whole experiment plant. And thank you very much for your kind attentions.